Good day. I welcome everyone to the seminar Did Life Originate from Low Temperature Areas of the Universe? This presentation has two parts. In the first part of my presentation, I will describe the processes occurring in space and show that the molecules formed in these low temperature areas are likely delivered to the planets and therefore can considerably contribute to their chemistry. And in the second part, I will show our recent laboratory studies that demonstrate that condensation of atomic gas which present in the SM leads to the formation of the variety of complex organic and important biological molecules, including even proteins. So let us come to the first part. Here you can see the life cycle of the matter in the interstellar medium. All heavy elements, heavy means heavier than lithium, are produced by dying stars here, and then they are expelled into the SM, where they have a chance to condense and form the molecular cloud, and the molecular cloud may further condense with increasing their densities until the star would be formed inside. After a star is formed, it has its own life cycle, and when this cycle would come to the end, the star would start to produce heavy elements. So all the cycle will repeat. In this life cycle, the very interesting and important object is a dense molecular cloud. Let's have a look on this object closer. Here you could see the picture of the dark molecular clouds. It is mainly composed from the dust particles, which completely block all visible light. Therefore, the temperature in these areas could be quite low, and the temperatures around 10 K are rather common. This object is very important because exactly here new stars are formed. And when new star is formed, it accretes all the dust which present here in the close vicinity, and protoplanetary disk would be formed. And this disk would further develop, and we have the formation of planetary system. And in the planetary system, we have a very small area where the life could exist, which is called habitable zone. It is simply defined by the temperature conditions which are required to have liquid water. But actually, no volatiles could condense in the habitable zone during the planet formation, and this would mean that we shouldn't have any liquid water on Earth. But we do have it, and explanation could be that volatiles are delivered later on the board of meteorites and comets. And asteroids and comets comes from the distant zones such as Kuiper belt and Oort cloud, and objects at these distant zones are very little processed by the light of the star, and therefore they expect to maintain original molecular composition of the dark molecular cloud. And if we investigate the meteorites which came nowadays on Earth, we can find a lot of important biological molecules. For example, almost all amino acids and nucleic bases were found in the meteorites. Also, many sugars and lipids were found there. Of course, such molecules could considerably contribute to the origin of life, and it becomes important to understand what kind of molecules are present in the dark molecular clouds and which molecules could be later delivered to early Earth and contribute to the origin of life. One of the common approach to detect any molecules in space is to perform spectroscopy. Then by comparison observational and laboratory spectra, we could identify any molecule. And this approach is widely used. And here you could see the table of the molecules which are currently detected in space. It's probably not up to date, but the main thing what I want to show here is that the number of the detected molecules is quite quickly decreasing with increasing of their size. And this could be easily understood considering that abundances of the molecules is also decreasing almost exponentially with increasing of the number of atoms in the molecule. But this would also mean that for the most interesting biologically important molecules which are big, the abundance would be very low and we would not be able to detect them by spectroscopy. Moreover, most of the detected molecules are detected in the gas phase and we know almost nothing about the solid state. To know more, we could use a different approach. We could also study the chemical processes which are occurring at the conditions very similar to those present in those dark molecular clouds, and based on the study, conclude about what molecules can be formed here. So these molecules could be formed either by the gas phase chemistry or by due to the surface reaction. And here I would like to show you that the surface reactions are much more promising for the formation of large biological molecules. And this is because on the surface, the associative reaction of the type A plus B given IB are allowed. A good example is the formation of molecular hydrogen, which can proceed on the surface but doesn't happen in the gas phase. And we can understand why is it by looking on this potentially energy surface of the interaction of two hydrogen molecules. So if you bring our system in this position, so in the position when two hydrogen atoms could feel attraction, of course, they will attract and we would have the formation of molecular hydrogen. But in the gas phase, the reaction energy cannot be dissipated, so the system will not stop here, it will go up, and then the hydrogen atoms will repel each other, and we would have a complete back dissociation, so the reaction would not take place. The situation changes when we have a third body, which I show here artistically as some liquid, but it doesn't matter. So in this case, the system would come to this position, the molecular hydrogen would form, the energy would be lost by dissipation to the third body, and the chemistry would lead to the formation of H2 molecules. In this case, the third body could be anything beside two H atoms. It could be any third atom, any third molecule. It could be the surface of the dust grain, or we quite commonly use in our experiment liquid helium, which is the most inert third body which you could only imagine. We should also consider what kind of surface reaction could be important for the formation of biologically and organic molecules. In this case, the carbon element is of the main importance, of course, and we should also consider that supernova explosions defines the initial state of the matter in the SM in the form of ionized atomic gas. And when the condensation starts, this ion gas could be quite quickly neutralized, and we have the formation of carbon atoms. But after this, we have two possible pathways. Carbon atoms could be converted into CO molecules in the gas phase, and then CO molecules can accrete on the surface of the dust grains. Or carbon atoms could directly accrete on the surface of the dust grains. And if we compare the times which are required for the conversion to CO molecules and the times which are required to condense on the surface, freeze out time, we can see that these times are quite similar. So we can conclude from this that quite large portion of carbonaceous material is formed due to the accretion of carbon atoms, and then reaction of carbon atoms on the surface basically defines the composition of solid carbon. But till recently, the reaction of carbon atoms on the surface were almost unexplored, and a probable reason for this was that it was very difficult to produce single carbon atoms, which are suitable for chemical studies. So to overcome this problem, we had to develop our own source of atomic carbon. And the design of this source you could see here. It's very simple. You have a tantalum tube which is filled with a carbon powder. Then this tube is clamped on both sides. It's inserted between two electrodes and heated by direct electric current. When the temperature increases up to 2000 K, the carbon solvated in the tantalum diffuses to the outer surface of the tube, from which it evaporates in the form of single carbon atoms. And here you could see the mass spectrometry study of our source here in comparison with the thermal evaporation of graphite rod. When the graphite rod is evaporated, we can see the formation in the gas phase of mainly carbon molecules, C3. In the case of our source, we can see the formation of carbon atoms with a few helium atoms attached. This is just because we use for the analysis the helium droplet experimental technique, which I will explain later. And we can see few carbon molecules, but these molecules were also produced not by our source, but were formed already inside of the helium droplets. So the source really produces only carbon atoms which have very low energy. So they are very suitable for chemical studies. And when we had our source, the first study which we try is to see what kind of carbon material would be formed by the condensation of carbon atoms with other most abundant molecules which are present in space. So we have helium droplet which flies in the high vacuum chamber, and then we dope these helium droplets with carbon atoms, H2, H2O, and CO molecules. And then we insert the substrate into the path of the helium droplet beam, and all refractory material should stay on the substrate, and we could analyze it. But to our surprise, instead of the formation of amorphous carbon or diamond, as we at that time also expected, we saw the formation of refractory organic material, which is evidenced by this infrared spectrum. And it contains also quite a high fraction of oxygen, which is about 25% by its mass, which is evidenced by EDX spectroscopy. So this study showed that already at this very low temperature of helium droplet, which is 0.37 K, carbon atoms react with all these molecules, and this reactions leads to the formation of the organic. At that time, we were unable to follow the pathway of the chemical reaction, and we just use a quantum chemical computation to suggest the following pathway of the organic formation. So to overcome this technical problem and to be able experimentally follow the pathway of chemical reaction, we developed the experimental technique, which I can demonstrate you with this video. So we have formation of helium droplet in the vacuum chamber. It flies, and then in the first pickup chamber, it's pick up the first reactant, and then it passes through the pinhole and arrive to the next pickup chamber. where it can be docked with the second reactant by the collision. When the second reactant is picked up, they can meet inside of the helium droplet and react. And then the reaction energy will transfer it to the liquid helium, which would lead to the evaporation of some number of helium atoms and to size reduction of the helium droplet. And finally, the helium droplet will arrive to the last chamber, where the size of those helium droplets could be investigated. And as evaporation of each helium droplet takes away about the same amount of energy, then knowing the size of helium droplet before and after the reaction, we can judge about the amount of energy which is released in this chemical reaction. So knowing this value, we could go further on and perform the quantum chemical calculation of the reaction. As an example, here I show you the energy level diagram of the reaction of carbon atoms and ammonia molecules. Here all levels correspond to specific possible product of this reaction. And of course, from this computation itself, we don't know what kind of product would be formed, because here we have a number of the transition state over which reaction should proceed in order the next molecules can be formed. And if the energy dissipation would be fast enough, the reaction can be frozen at different stages. For example, it could be frozen already at these weakly bounded complexes. So then, if we perform the measurement of this reaction, we got this number of energy which is released in this reaction, and we can compare with this energy level diagram. We know that there is a good match with this level, and we consequently can expect that this molecules is formed in our reaction. We apply this method to study a number of reactions of carbon atoms with small abundant molecules present in space. And currently there is a very interesting picture. Here on the left, you could see the molecular composition of molecular ice mantles. And on the right, you can see the number of the reaction of carbon atoms towards these molecules. And with a blue font, I mark those reactions which are fast at low temperature. And as you may see, there is only one exception. The carbon atoms doesn't react fast with methane molecule. And this result have several very interesting consequences. First, carbon atom don't need to diffuse on the surface to react with other molecules. When carbon atoms land on the surface of ice mantle, it will react with the first molecules it will meet. And this is not considered in many chemical models. The second conclusion is that this is, of course, a new pathway to the formation of organic molecules, because the product of this reaction in many cases is organic molecule. And finally, such reaction of carbon atoms lead to the release of really big amount of energy. And this may have several consequences. First, it could help to desorb the molecules from the solid into the gas phase. And it could also promote the chain reactions. The chain reaction is a reaction when some reaction is promoted by the energy from which is released in the previous reaction. Such an example of the chain reaction you can see here. When carbon atoms react with H2 molecules, it leads to the formation of HCH molecule. and it's followed by the release of about 24,000 weight number of energy. And then HCH molecules can react with ammonia and CO2 molecules, leading to the formation of glycine. In the singlet channel, this reaction is completely barrierless, but in the triplet channel, there is a small energy barrier, which could be in principle simply overcome due to the energy which is released in this reaction. Or otherwise, this energy could promote the system into the singlet state where this reaction pathway is completely barrierless. We also investigate this reaction in experiment. We doped helium droplets with ammonia, CO2, and H2 molecules, grow the ice cluster inside, and then add carbon atoms to this ice cluster. And the effect of addition of carbon atoms to this ice cluster you could see in this mass spectrum. This mass spectra show the formation of the peaks which belong to the glycine. So, and it supports the feasibility of this pathway of the glycine formation. The other pathway to the glycine could be initiated by the reaction of carbon atoms and ammonia molecule. This reaction leads to the formation of this molecule, and these molecules could also participate in this reaction, which have some energy barrier and would lead to the formation of glycine. So there are different possibilities of the glycine formation, but it's not very new because the formation of amino acid at the conditions of the SM were already demonstrated many times. And we just think whether we could make a next step towards the origin of life and see whether we could form a biopolymers here. But formation of proteins happens in the following way. So two amino acids here meet together, and they need to detach those parts which are marked here with a blue color. Basically detach water molecule. And because you need to break some chemical bonds, this reaction has quite high energy barrier, and it doesn't proceed even at room temperature. It proceeds either at elevated temperature or action of some specific catalyst is required. But interestingly, if we look in this reaction pathway, we can see that in this reaction pathway, the highest energy barrier is associated with addition of water molecules, and then we have a quite high energy barrier to detach water molecule. And this road then doesn't look very reasonable. We have two very energy unfavorable steps. And of course, the idea was whether we could skip the stage of amino acid formation and go directly to the proteins. And our quantum chemical computation shows that indeed the formation of aminoketene could be barrierless in space. And starting from this aminoketene, we can very simply build the protein chains, as you can see in this video. We decided to test experimentally whether it is possible to form aminoketene in our experiments. We use experimental setup, which is shown here on the figure. We have the substrate, which could be cooled down down to 10 K, and what's going on the substrate we could monitor by means of infrared spectroscopy, and what's going on in the gas phase we could monitor with a quadrupole mass spectrometry. And then we deposit ammonia CO gas and carbon atoms on the substrate. Here you can see the infrared spectra of the deposit. The red spectrum is obtained after we deposited only ammonia and CO molecules, and the black spectrum is obtained when we added carbon atoms. As you may see, the addition of carbon atoms led to the appearance of a number of new absorption bands, which shows that reaction proceeded 10K, and we have the formation of new functional groups like CCO, CC, CN, and considering the presence of these functional groups and the initial reactants, we could only conclude about the formation of aminoketene at 10K. Later, the formation of aminoketene was also confirmed by the quadruple mass spectrometry. When we start to hit our substrate, we could see the desorption of these molecules close to 300K. But more interesting stuff happens when we started slowly rise the temperature of our substrate. When ammonia starts to evaporate, we can also see the decrease of spectral bands which belong to aminoketene molecules, and we see appearance of the number of new bands which are marked here by the dashed lines. And you can see them better on this right image, where the deconvolution of this room temperature 300K spectrum is given. Here the most informative is appearance of new bands which are marked here peptide 1 and peptide 2. These bands are characteristic bands of the peptide bond, which link to amino acid in the protein chains. Peptide 1 is CO stretching of this group, and peptide 2 band is more complex. It is composed from mainly two vibrations. It is CN stretching vibration and NH bending vibration. As peptide bond is not present in the aminoketene molecules, we can conclude that infrared spectroscopy tells us that with increasing of the temperature, aminoketene polymerize, forming at least diamonds. But of course, at this stage, we don't know what kind of material is produced. So, to know more about the produced material, we extracted the substrate from our ultra-high vacuum chamber and recorded its infrared spectra. We saw that material was not affected by exposure to air, and consequently we can do the further analysis. This applied the substrate to the quadruple mass to orbital mass spectrometry, which is equipped with the electrospray ionization method. So the material was weighted in the water methanol mixture, and the obtained mass spectra you can see here. There are a big number of the bands, but the important thing is that most of the bands, at least which are marked here with the red color, belong to a series or to progression, where the spacing between the bands always correspond to the mass of monomeric molecule aminoketene. And the presence of this progression again tells us about the polymerization of aminoketene. So the result of mass spectrometry confirms the result of infrared spectroscopy and tell us that there is a polymer formation. And we can see the addition of up to 8 or even 9 and 10 monomeric unit in this chain. For some of this progression, we were able to understand the molecular structure. For example, the most intense progression, which starts from this peak, corresponds to polyglycine, which is terminated by 2 amino groups at both ends. Just to be absolutely sure, in our assignment, we also perform the collision-induced dissociation of this ion. So we use a quadrupole mass filter to preselect this ion. Then these ions were sent to the collision cells where they collide with argon atoms at about 30 eV energies. And here you could see the fragmentation pattern of this ion. And the fragmentation really correspond to the predicted molecular structure. So we can be sure about our assignment. So, and this result is quite well in line with a recent potential detection of protein in meteorites. Why I say potential? Because although the proteins in meteorites were found, also were unable to confirm that these proteins came from space and not terrestrial contamination. But as you may understand, it's not so simple. But in combination with our result, this detection of proteins becomes more confident. And finally, there is very interesting study of amino-terminated polyglycine molecules, which were synthesized by the complex chemistry process. These amino-terminated polyglycine were most abundantly formed also in our experiment, and they were found to be capable of very efficient self-organization. So they organize themselves in the way, as you may see on these figures. And as to me, it's very closely resembles the way how the molecules in the cell membranes are organized. And this, of course, opens very interesting opportunity for the origin of life. But yeah, at this stage, it's too early to say about anything definite. It's still a bit of speculation, and more studies would be required to understand more. And I just want to conclude my presentation that we performed a study which shown that chemical process occurring in space at low temperature can lead to the appearance of sufficiently large organic and biological molecules, including proteins, and then they're expected to contribute to the origin of life on the planets. And the found non-energetic pathway of protein formation could have an advantage for the survival of the formed molecules. Since if their formation would take place in the areas with low UV flux, the molecules can be buried in the bulk material, and this bulk material will protect them from further destruction. And I should also admit that currently this low temperature chemical reaction of carbon atoms are almost unexplored, and we need more studies to understand better the chemistry. And I would like to thank you all for your attention. And I would be ready to answer your questions.
Many thanks, Serge. For asking questions, you have again two possibilities. Either you switch your camera on and I will give you the word to ask a question, or you can also use the chat forum for asking a question. So these two possibilities you have. So the floor is yours.
Hello?
Mario Acola, you wanted to ask a question?
Yes, I have a question. Can I make this question?
Yes, please.
So maybe I lost some first slide, but I want to know which is the mechanism and the setup that is devoted to the formation of atomic carbon.
You mean in the experiment or in space?
No, in the experiment, the experimental setup you use to form this atomic carbon.
Okay, I should go back. I have a slide.
Sorry, maybe I lost some slide. You already.
No, it would be nice, yeah, to tell a bit more.
Thanks.
Okay, can you still see my screen?
Yes.
Okay, so what we do, we have a tantalum tube here. It is filled with carbon powder. When we fill it with a carbon powder, we clamp its end, and it's placed between the electrodes, and it's heated by the direct electric current. Then carbon basically is solvated in the tantalum and diffused to the outer surface. And when it's evaporated already from the surface of this tube, it's evaporated in the form of carbon atoms and not carbon molecules, as it happens when you evaporate carbon from the surface of the graphite rod.
Okay, thanks.
Yeah. Okay, good.
Okay. There is a question via the chat from Sebastian Quellner. Maybe I overheard it, but could you maybe please emphasize if your findings contribute slowly towards grain surface process, or also could be taking a role in the ISM? So far as I know, as grain surface processes are quite important in the ISM, but I leave the question to you, Sergei.
Okay, yes, this reaction is expected to take place on the surface of the dust grains which are present in the SM in these dark molecular clouds. And yes, in this sense, it should be relevant to the process occurring in the SM. And basically, just to answer a bit more, this dark molecular cloud is composed from the dust particles. So basically, what is blocking here, the all visible light, is dust particles. So there are many dust particles present there, and the process occurring on its surface are very important for the chemistry of the ISM.
There is another question via the chat from Ruth Sophie Taubner. Maybe I missed it, but as far as I know, the detection of glycine in the ISM was highly debated. Any news about that?
As far as I know, there is no glycine detection in the ISM. So, and yeah, as I explained, if you look in this table of the molecules which are detected in the SM, with the increasing of the molecular size, the abundances of the molecules are dropping down exponentially. And of course, there is no doubt that glycine is present in the ISM. It has been detected in the comets, so for sure it is there, but their abundances probably are so low that we cannot detect it. But it's applied to the.
There was this.
Sorry?
Yes, there was this claim detection by Steve Charnley, which was already, if I'm not mistaken, which was already in the 90s, where he claimed actually to find some lines, and this was then disputed by Snyder. And I think there has been some newer research that seems a little bit to vindicate the detection, but I don't really know what is actually the latest news about that.
Okay. I think it is still under the question, but yeah, as I said, for sure, the glycine should be present, at least in the solid state, in quite considerable amounts, as it was also detected in comets. But in the gas phase, probably abundance is not so high that it could be really easily detected.